Hello everyone, I am just going to review translation and transcription to help you with the whole process. Now I started with this first image because I like that it kind of talks about the different parts. So replication is when DNA is replicated. So you have your double strand, your double helix, you have your sides which are made up of phosphates and sugars, and then you have all those bases in the inside. So you have those bases of adenine, thymine, guanine, and cytosine. So you have all these bases inside. So what happens is, again, to review everything, is you have your helicase, which helps to unwind my DNA. So it's going to just unzip it, open it up. So now I have my unzipped and I have all my bases. So what will happen then is it's going to add on basically because of base pairing. So A goes with T, C goes with G, and it just is going to complement it and match those base pairs. And this is going to unzip it, so the new stuff is going to be on the very inside and the old stuff is going to be on the outside. So when you end up, you're going to have two new DNA, and they're going to be one old and one new strand. So this is then my old and new, and that is my DNA. But I cannot forget my outside, which is my sugar and my phosphates. That's DNA replication. If you look at the other parts of this document, they have transcription. So transcription is, if you think about just the etymology of it, script means to write. So you're rewriting DNA. DNA can't leave, it's too large. And it has so much information, it's just going to take a portion of it, rewrite it, just what it needs, and take that out of the protected nucleus. And that is our mRNA. Now RNA polymerase helps make our mRNA, and that's the whole process of transcription. It is just a copy of DNA, a portion. Then the last part is taking this and translating what those letters mean into our protein, which is basically a chain of amino acids. And how it does it, 3 at a time, look at each codon, and from there match it up with then an amino acid. So this is what we looked at yesterday. So we have our nucleus, which holds our DNA. This is, again, the first part, transcription. It's going to make a copy of the gene that it needs to make. And that is our mRNA, which is that copy which leaves through a nuclear pore into our cytoplasm to start the next step of translation. Translation is where it's going to read 3 at a time and translate the code. It's going to look at a codon found on mRNA. It's going to match up with the tRNA, which is going to transfer that amino acid. So this has the anticodon. So again, if my codon is A, U, G, A goes with U, U goes with A, G goes with C. So let's pretend this matches, this has that code on it. It's going to match up with those, releasing an amino acid. Then this 3 will be red, match up with the anticodon. This will then be added on to that chain of proteins. Again, a chain of proteins is then or a chain of amino acids is then our protein. So just to review, some of the RNAs are RNA. That is basically what makes up the ribosome, the site in which proteins are made, so where proteins are made. Our mRNA is a copy of DNA, and our tRNA, this contains that anticodon, which is going to match up with the codon and transfer amino acids. Those are the jobs of our RNAs. So I like to think of this as like mRNA is the recipe, because it's a copy of the original. RNA is where things are put together, so it's like the kitchen. And the tRNA is what kind of reads that code and then puts it all together. So that would be like me in the kitchen. Make sure you study. Good luck. If you have questions, please see me.